Hello learners, welcome to our ongoing series on photosynthesis. Till now, in the past two lectures, we have traced the path of history and the development of concept of the whole equation of photosynthesis. In the second lecture, we stressed upon various types of pigments that were responsible for functioning in the whole process. And we had asked ourselves a few questions as to why has nature chosen chlorophyll A and chlorophyll A alone as the principal photosynthetic pigment? Does it enjoy certain special privileges in terms of its properties? Yes. In this lecture, we would like to talk about the absorption and action spectra of different photosynthetic pigments, and then we would like to come to a conclusion why has nature chosen any pigment to lead the whole process. In addition, we will also look deeply towards what do we mean by light, its quality and various parameters. In the last lecture, we had left on a note that Hill's reaction and various other initial experiments had come to made us come to a conclusion that the acceptor of electrons, which was done in the in vitro conditions, that was done by benzoquinone, ferrocyanide, DCPIP in the laboratory and so on. But then what actually happens in nature? In nature, NADPH, which is the reduced coenzyme 2, nicotinamide adenine dinucleotide phosphate in a reduced form, is the actual acceptor of electrons, and then by this account it functions as a reducing agent. And therefore, before coming to the types of reactions which are involved in photosynthesis, let us find out what do we understand by the term Q10, which is a very important term in biochemistry. Actually, Q10, which is called as the temperature coefficient, is a ratio between the reaction rate at say T plus 10 degrees to the reaction rate at 10 degrees. What we mean is that if we increase the temperature of a reaction by 10 degrees, what is the effect on this process? We find that for physical processes, the Q10 would be say more than 1, 1.2, 1.4. And for enzymatic reactions, for example, we will see in the dark reaction, that is the Kelvin cycle, or in all the reactions of aerobic respiration and anaerobic respiration, these are all physiological processes, the range usually goes from 2 to 3. That means it is a multitude. But then when we talk about a photochemical reaction, then surprisingly the Q10 is 1. Learners, you should remember, whether you are talking about the formation of chlorine, whether you are talking about the formation of ozone in nature or the formation of nitrous oxide, these are all photochemical reactions. These are governed by light and therefore they are insensitive to temperature. There were various intermittent light experiments that were performed by Warburg and then later by two scientists, Emerson and Arnold. And if you remember, learners, Emerson was also a Nobel laureate. They actually demonstrated that photosynthesis consists of two different phases. The first phase is a very rapid process whose Q10 is 1, and we agree that the phase 1 is actual photochemical reaction, which we later called it as the light reaction, and then it gives rise to a product which is then very slowly converted into the final product C. And you know what the final product is. In this case, it is the hexose sugars. And the second phase, which is a slow phase, is a thermochemical reaction. It is very sensitive to temperature and it is light insensitive. And then it was called as dark reaction. You remember we had raised a question in one of our earlier lectures that dark reaction, does it occur only in dark? I said no. Dark reaction occurs in light as well as dark. It is not sensitive to light, but then it is thermosensitive. So, this was the basic set of experiments which laid down the foundations of light and dark reactions. So, that means to summarize, we have two stages of photosynthesis, and the products of light reaction are going to be ATP and NADPH. When we are talking about the cyclic and the non-cyclic photophosphorylation, we will have ample time to look at these processes and their formation in details. And then this light reaction is the one which drives the subsequent stage, and that is the dark reaction. And as you know, learners, dark reaction is another name for the C3 or the Benson-Calvin cycle, where these two products, namely ATP and NADPH, are utilized for the formation of sugars. This diagram which you see on your screens now is a summary diagram which would need to be repeated for one or two more lectures, because our discussion is going to revolve around these key processes of light reaction and the dark reaction. But just to take a small fraction of it, you can see on the top light being bombarded on chlorophyll molecule, and in the next step water seems to be broken down into OH and H ions, and as a result molecular oxygen is being released. Now, this process is taking place in the grana. Imagine the process has hardly begun and oxygen has been released. It gives us a very important thought, and that is the evolution of oxygen the sole aim of photosynthesis? Certainly not. The aim is to produce sugars. The aim is to convert the solar energy into potential chemical energy in the form of sugars which can later be respired. But then why did oxygen be released? This is one of the most fascinating coincidences in nature, which is keeping the entire aerobic life possible. If water was not taken as the ingredient, as the basic raw material, oxygen would never have been released. So, that means the choice of water was through natural selection, which gave rise to oxygen. There are so many bacteria which utilize H2S. They would never release molecular oxygen, they would release sulfur. So, that was the basic difference between oxygenic and anoxygenic photosynthesis that we had talked about in our earlier lectures. So, therefore, coming once again to light reaction, the rationale would be to release oxygen, ATP and NADPH. Of course, oxygen would be a byproduct, but then as far as the remaining two is concerned, they would be responsible for the formation of sugars. Since we are talking about the role of light, we know that in this light reaction, which is a very rapid one, we have three different phases: photoexcitation, electron transport and photophosphorylation. So, the first and the foremost step is that at least the light should first be absorbed by a photosensitive molecule, and that happens to be chlorophyll. And in the process, this use of solar energy would be utilized for splitting water and the subsequent release of electrons. So, when we are talking about photoexcitation, first we should know from where is this radiation coming from. Now, this whole discussion involves a little concept of physics, because when we know that the total solar energy which is generated by sun, enormous amounts, just 40% of it is being bombarded on earth. So, it finally reaches earth, and it has been calculated that it is about 5 into 10 to the power 20 kilocalories. Now, lots and lots of it is dissipated. There are various gases which scattered, and in itself light is a particle of radiant energy, which we call an energy packet or a photon. And if you look at your screens a distribution pattern in the form of a graph, we find that out of the total light which is falling, some of it is absorbed, some is transmitted, and a little form is also reflected. But if you look at this graph carefully, much of the visible light is actually absorbed, and it is this visible light which is running the show of photosynthesis. But then you have so many other wavelengths in addition to visible light. So, that means there is a very, very broad range. How do we distinguish them? We will distinguish them on the basis of their frequency, which is denoted by V, and of course, by lambda, which is the wavelength or the wave character. If you look on your screens very carefully in electromagnetic spectrum, you would find a whole range coming from left to right, and that is of the cosmic rays, the X-rays, then the beta and the gamma rays, and then the UV rays, and a very, very thin band of visible spectrum, which can later be resolved into 7 colors, the Vibgyor colors, and then passing on to infrared and the microwaves and the FM and AM and the other radio waves. But the point is, one point that you have to realize is, the more on the left you are, that means you are talking about high energy radiations, they have a very high frequency and a low wavelength. The moment you turn more towards right, you have a higher wavelength, low frequency and less energy, which gives us the idea that wavelength and frequency are two terms which are inversely related. Therefore, the short wavelengths have higher energy. On a very light note, if you were to give your grandfather a gift to soothe his aching muscles and bones, you would give him an infrared lamp, which would produce enough heat to soothe him. You cannot give him a UV lamp, because if you look at the screens, UV would have enormous amounts of energy, it would burn everything. So, that means out of the entire energy spectrum, just the visible light is one which interests us, because our eyes are sensitive only to this very narrow zone, say, which is ranging between 400, that is violet, and 700 nanometers, which is red. So, this particular portion is actually photosynthetically active, and therefore is called as PAR, or photosynthetically active radiation, because this is the effective region for photosynthesis. And if you were to look on your screens carefully, there is a very dark band of violet which is on the left hand side, right. So, to summarize the whole aspect, we can say that light is coming in the form of energy of different wavelengths. Shorter the wavelength, greater would be the energy, and it would be able to excite the electrons of chlorophyll to a higher level. And certain wavelengths of light, they are detectable by human eyes, and they are seen as colors, because we cannot see all the light. Talking of colors now, we talk about the concept of absorption and action spectrum. Let us ask a question, which is a very foolish question to a common man, but then which has a very serious answer. Why are all the plants of the world green? Why is not the entire vegetation red? The creator could have made them red, because red is also a color of the visible spectrum. Well, a common man would say that the plants or the leaves are green because they are reflecting green, very true, but then the answer is only partial. So, let us see what do we find an answer in a scientific manner. We are talking about the different types of energy levels which are created by these light packets. Blue light, for example, has a very high energy value, it is about 274 kilojoules per mole. On the other hand, if you see carefully, red light is only 181 kilojoules, so it is less. And there exists a relationship between the number of photons and of a particular wavelength which are going to excite. Chlorophyll is not selective that way. All the wavelengths of photons are going to fall on it. How much can it absorb will depend on the energy values. So, that means the first step in this process is a photobiological process, that is how much and what is the chlorophyll molecule absorbing in terms of packets of light quanta. So, that means this degree of absorption can be easily seen and recorded with the help of a spectrophotometer. Coming to my basic question, why do all these pigments, why do the plants, they show different colors? The answer is simple, because if we make a graph with the help of studying the absorption through a spectrophotometer, the color of the pigments will be due to the wavelengths of light which are reflecting back. A leaf is looking green because it absorbs some other wavelengths and reflects green. Likewise, for a red color. So, this whole graph, which is depicting the absorption at different levels with respect to the function of wavelength, is called as absorption spectrum. Mind you, absorption spectrum is like a key to the identification. If we are a little more simplistic, we can even say that it is just like a fingerprint, which is very, very specific for a given wavelength. So, the absorption spectrum of chlorophyll is different, because we know we had seen it in our second lecture that it absorbs mostly blue and red wavelengths, and therefore, it reflects green. On your screens you would find that the color of the pigments is different, and the absorption of their wavelengths would be entirely different. Now, this is a very interesting ramification, because you would find that in a marine habitat or in aquatic habitat, the green algae seem to be on the surface. When you go a little deeper, brown algae exists, and the deepest in the photic zone, wherever light can penetrate, you have red algae. Why is it that there is a zonation of algae or of the macrophytes of different colors? Now, I think you can understand the phenomenon better. The Russian scientist by the name Gaidukov gave the principle of chromatic variation and the chromatic adaptation. He said the blue wavelength has the highest energy level because it has the shortest wavelength and therefore, the highest frequency. Now, it can penetrate deeper. It is a matter of common sense that an alga or a plant which is absorbing blue wavelength cannot reflect blue. It will have to reflect a wavelength which is longer than the one it had absorbed, and therefore, it would emit red light. That is why it will appear red. The same logic can be given for brown algae and for green algae, because red light is the weakest as far as the penetration is concerned, and therefore, the ones which are absorbing green, like you see all the plants on earth or most of the plants, they are getting most of red light, and therefore, they would appear to be green. So, we have on display the absorption spectrum of carotenoids, which is different, and what is important is, if we look at the comparative study of these different absorption spectra, we would find that they have different peaks in the different light zones. We find that chlorophylls, since we are interested in chlorophylls, they are the main pigments, they absorb light mainly, as I said before, in the violet blue, and there is a very, very thick band of violet blue, which is called as the Sauret band. Now, the Sauret band would signify that it is full of lots and lots of high energy particles of photons. The important point is that merely by looking at the absorption spectrum is not enough, because every pigment will absorb one wavelength or the other. It is very natural, but then it is important at which wavelength it is actually working. So, the relative activity of a particular physiological process would be at a particular wavelength, and the graph of which is called as action spectrum. Now, one of the other very, very interesting coincidences in nature has been that chlorophyll A matches its absorption and action spectrum to the highest level as compared to other pigments. Learners, if you have understood this logic, it means that whatever little wavelength is available to chlorophyll A, it is the most efficiently utilized. This perhaps gives us an answer to the foolish question that we had asked a little earlier as to why most of the plants on our planet appear to be green, because they have chlorophyll A, and chlorophyll A therefore is the principal pigment. Now, let us go a little deeper into the physics of the whole process of photoexcitation. We know that when the photons are going to be absorbed, chlorophyll is going to be in different stages of excitation. If there is no excitation, then a molecule is in its ground state. And the moment absorption is there, it is a very, very rapid process of absorption. You can find it is unimaginable. It is about 1 femtosecond, which is 10 to the power minus 15 seconds. So, that means the photoexcitation is such a very, very quick process, and we find that it is not just one electron which is performing a photochemical change. It is a complex molecule with so many electrons, and each electron has to absorb photons of different wavelengths, because we are not talking about just red or blue, we are talking about all the wavelengths, because the other accessory pigments also have to be involved in it. So, the electrons after excitation are going to be elevated to different excitation levels, which are called as singlet stages. So, blue light is having a very high induction effect as far as the excitation energy level is concerned, and anything which is going to be exposed to blue light would have something like 58,000 calories per Einstein as compared to red light. Incidentally, 1 Einstein happens to be 6.03 into 10 to the power 23 photons. So, what we find is when blue light is falling on chlorophyll, chlorophyll molecule electrons are ejected to a much more excited to a much higher level, which is called as the second excited state. When they are now not reacting with any process, they have to come back. So, they will come back to the singlet stage, but it is a very fast process, because they are highly unstable, and their half life is only 10 to the power 12 seconds, which is just 1 picosecond. On the other hand, if the exposure is to red light to the chlorophyll, then it has a slightly lower energy, as we saw, and therefore, the electrons can be ejected only to the first excited stage, which is the singlet stage, and there also it is an unstable one and short lived, however, less short lived, it is 1 nanosecond. So, the loss of energy when these excited electrons are coming down have to be released in one form or the other, because the first law of thermodynamics clearly states that the energy, whether it is created, can neither be created nor destroyed, but still it has to change its form. So, there are 4 ways by which extra energy is lost. Number 1, as heat, as light, as inductive resonance, and as photochemical reactions. The graph on your screen clearly summarizes that if the excitation was through blue, then we find that the second singlet stage is achieved, and then it will gradually come down. That is why the absorption of blue wavelength would result in a release of red fluorescence. That means a change in its wavelength pattern. And if absorption is by red light, then the excitation itself is to a lower level, that is to a singlet stage. So, this energy loss as light is called as fluorescence. So, that means fluorescence is nothing but re-emission of photons as light by their electron decay. So, whenever there is a excitation, fluorescence will take place, and fluorescence would always go to the right hand side, which means from a higher energy to a lower energy. That means alternatively we can always state from a shorter wavelength to a higher wavelength, and this is called as Stokes effect, which is named after its discoverer. The other form would be that there is a resonance transfer. That means resonance transfer is what is called as the radiationless transfer. This is very important among the chlorophyll molecules, the antenna molecules, and here it is the sheer proximity between the two light sensitive molecules, which are called as chromophores. And the other possibility is a photochemical reaction, because coming from the second triplet stage, the electrons may pause to a transitory stage, which is called as metastable triplet stage, which is long lived. Because you will realize, learners, a reaction requires a certain time. It requires some time for the electron to be stable. So, that means this is a long lived stage, say approximately 10 to the power minus 3 seconds, and this is enough to cause a photochemical change, and also the radiation of wavelengths, which is called as phosphorescence. So, if you see a summary diagram, you would find that there are various levels of excitation depending upon what the wavelength was. So, when the photochemical reaction is given ample time, we find that emission of heat, emission of light is really not important, is really not useful for photosynthesis. So, what is useful for photosynthesis? The photochemical reaction, which is the best alternative, because it gives you most useful reaction to initiate the process of photosynthesis. And it is this photochemical reaction which now excites the antenna molecules and the reaction center. So, that means photoexcitation would mean that a chlorophyll molecule is excited. When it comes down, if it reacts with some other product, fine. If it does not, it has to come back. So, from an high energy state, it comes to the ground level. What is going to happen to the extra amount of energy that it is dissipating? It could be in the form of light, in the form of heat, but as I said earlier, they are not useful to us or to a plant for that matter, and it is only a photochemical reaction which it is going to initiate at the metastable state, which is going to run the show of photosynthesis. In our next lecture, we will talk about the discovery and the intricacies of the pigment systems, which are now called as the light harvesting centers. Thank you.